Okay, so let's start. So good morning, everyone, and I think we have a good audience today for this edition of the bioinformatics seminars. As you know, this is organized by the students group in bioinformatics, and also the our group, research group on bioinformatics and systems biology, which is a part of the Center in Biological Engineering. So I welcome you all to this session. And first of all, I want to also thank Dick the Reader, which accepted our invitation to speak about his work at Wageningen University and research. So thank you, Dick, for accepting our invitation. I think you have two stories to tell us about data-driven bioinformatics. So I'll pass you the floor, and thank you for accepting again.
Okay, thank you very much, Miguel, for the introduction and also for the invitation. I think that COVID-19 doesn't make our life a lot easier, but at least it makes it easier to do seminars like this and to exchange ideas like this. I think we're getting used to that, and that's one thing one good thing maybe that can come out of this whole situation, that we find it easier to connect in this way. So as Miguel already said, my name is Dick de Ridder. I work in the bioinformatics group at Wageningen University and Research, and the title of my talk is a bit bland, a bit generic, and that's because I would like to share with you two stories which are quite different, and I couldn't really come up with a good title that called both of them. But I'll start off by briefly introducing the university and my group. So the university is located in Wageningen, which is actually a small village somewhere in the center of the Netherlands. And it's one of the smallest universities. And it's quite specialized. In contrast to most other universities, we have a sort of specific a number of topics that we work on, and they all have to do with health, lifestyle, the living environment, and development. So really, the life sciences and the environmental sciences. That's our focus. The mission of the university is to explore the potential of nature to improve the quality of life. So it really has these two aspects, that we want to do fundamental work, but we also wanted to find application. And one major element in that is that this end research at the end, I hope you can see my pointer, that's actually a contract research organization which is part of the university. So we have, well, basically part of the entire organization. So we have everything from fundamental research to people working on contract research with companies together, and everything in between. And that makes it really easy for us to get in touch with industry and other stakeholders and make sure that our work is applied. The bioinformatics group is shown here in a, well, not a very recent picture, because we couldn't get together like this over the last year anymore. But it's a group of, at the moment, 7 staff members and 15 to 20 PhD students and postdocs, and who focus on developing and applying computational methods, working mostly starting from omics data to get to biological knowledge, and mostly in the green life sciences. We sometimes say we work on anything except human. So we try to stay away from medical research, but everything else, so plants, microbes, fungi, microbiomes, etc., we work on. As I said, the group has 7 staff members. Here they are, each with different expertise. So you see a lot of omics, so that's where we find each other. But there's people that work more on transcriptomics, on specialized metabolism, metabolomics, microbiomes, and metagenomics, and so on. So we do have a shared interest, but we all have our different niches. And the story I will tell today is in collaboration with 2 of my staff members, Sandra Smit and Alt-Jan van Dijk, who work on comparative genomics and also on machine learning. My own research interest is in, I have more of a computer science background, so it's in how to represent, process, and analyze this omics data. And I like to often involve machine learning in that. And my main questions are then, how can we integrate prior knowledge and measurement data, which is often easier said than done? and how do we work with data from new measurement technology. For example, we're working together with physicists on developing devices to sequence proteins, so actually read off the sequence of proteins. Well, these are still very much on the drawing table. We don't have these devices yet, but we can already think about how can we simulate these, how can we model these devices, and what type of algorithms would we need to process the data that comes off. The examples that I want to discuss today, the two topics are pangenomics and protein structure-based prediction, which don't have a lot to do with each other. So I propose that for the first half of the talk, I'll speak about pangenomics. Then maybe I can take some questions on that first. So if you have those, then please put them in the chat. And then I'll move on to the protein structure-based prediction. So this pangenomics, that's basically motivated by the idea that we're now in a post-genomics era. So I think you probably all realize that we're swimming in genomics data, and not just raw data, but also draft and full genomes. These are a couple of screenshots that I took last week of NCBI, which lists here like a couple of 100,000 prokaryotes, 40,000 viruses, 15,000 eukaryotic genomes, and we get more of these each day. Also in the Joint Genome Initiative database, JGI Gold, you see these numbers really shooting up. So it seems like by post-genomics era, people indicate that measurement no longer is an issue. We have so much data. Really, I think that if you now write a proposal saying that you want to sequence something more, then I think the first question should be, can't you already use what is already out there, right? So analyzing the data now is really the main challenge. And the people that we collaborate with in Wageningen, they bring these challenges. They're interested in analyzing large collections of genomes of bacterial and fungal strains that are of interest, or of plant and animal species that are of interest. And in Wageningen, those are often very agricultural. So we have people working on tomato, potato, but also melon, pepper, on pigs, cows, and chicken. And also on pathogens and diseases. And so in all these areas, we see that people are sequencing not one genome, not two genomes, but now lots of different wild varieties, which may contain interesting new genes that you want to breed into your plants or animals, or of these cultivated varieties to learn how variation in these cultivars is related to the phenotypes we see. For example, getting smaller or larger tomatoes or tastier bacon. We try to link that to the variation we find. And there's also quite a few more fundamental scientists working on looking at genomes to find unique traits. For example, there's a couple of collaborations we have on these two plants. And now I'm really not an expert, so don't ask me too much about this. But this is a tree which is able to nodulate. That means basically it's able to attract bacteria to its roots. And it depends on these bacteria for its nitrogen cycle. And it's quite unique in that, so that if we understand that mechanism, then we can also try maybe to breed that into other crops that could really benefit from that and grow better. And this small plant here, it's called Hirschfeldia incana. This is a plant that grows around the Mediterranean, is known to be extremely good at photosynthesis, much better than many other plants. And so we're studying that also to learn maybe what copy number variation or genomic variation is responsible or underlies that phenotype. Right, so what is common in all these challenges is that there's a question, what happens in my genome? And now, I guess you know that the traditional approach is that if we sequence or resequence a number of individuals, and then we compare it to a reference genome. Now, in human genetics, this is quite useful. We think that the human reference genome, of course, it is a sort of a combination of a few humans and maybe not completely clear what human it represents, but at least it's fairly close to other human genomes, so that comparison makes sense. But in these plants and animals, it often starts to become a problem if these genomes are a bit further away, if they're further away in evolution, or if they've been heavily improved by breeding. Now then you lose a lot of information if you just compared to a single reference. So ideally, you would like to maybe get full genomes for all these varieties and then compare all these genomes to each other if you do comparative genomics. But that quickly becomes very costly, as you can imagine. Eh? So if you have 3 genomes, you need to do 6 comparisons. If you have 10 genomes, it runs into the tens or hundreds already. So this doesn't scale. So a solution that many people are working on is pangenomics, where we find, say, a single representation of an entire set of genomes that we can then do analyses on. And I think it's important to, I'll say that now, and I'll say it again a couple of times during the presentation. The goal here is not to do something that we cannot do yet. So I think we want to achieve, be able to perform analyses that we can already perform, but at a scale that we're not able to do yet. So you can imagine if we're able to use a pangenome to compare all genomes to each other at once, then maybe we can do something in a couple of hours that would ordinarily take days or weeks if we would do all versus all comparisons. But in principle, nothing changes. It's just the scale at which we can do it. Well, this pangenome concept is actually has been around for a while. It was coined in 2005 in a landmark paper by Terpene, where they use it to describe the gene repertoire of a species. So they sequenced back then a number of bacteria, and they found to their surprise that these bacteria had a sort of core genome, so a bunch of genes which were always there, which apparently needed to be there to be that species. And then an accessory genome, so sets of genes that were either present or not in subsets of bacteria, and even unique genes, so genes that were unique to individual subspecies. So this has been investigated further also in plants. People have made pangenomes of maize and rice and soybean, etc. And the same picture there emerges. You get these plots like these, where you see the number of genes, unique, oh, sorry, I shouldn't click. The number of unique genes goes up. I think this was a Brassica species, goes up from 56,000 to over 60,000 if you include more genomes. And the number of core genes actually goes down. So you find that there's a sort of core here, probably slightly less than 50,000 shared by all these plants. But each individual plant then has 10,000 additional genes, which can differ between plants. Well, this is an interesting way of looking at it, but it's fairly limited to only genes. So people have also started thinking, can we represent not just the genes, so gene sets, but also sequences in this way? And actually, my group already started working on that a while ago. So inspired by the Cortex Assembler, which I don't know whether you remember that, but that was an assembler which allowed assembling multiple species at once, multiple read sets at once, and then showed that in the resulting assembly graph, you could detect variation like this. So you could, for example, detect here things like repeats or polymorphisms as bubbles, or you could even see here sort of like shared regions by looking for shared paths in this graph. And so a while ago with a PhD student of mine, Jürgen Nijkamp, we came up with 2 ways of analyzing copy number variation and sequence variation in context by taking such a co-assembly approach, where we assemble 2 sequences together. We get a graph which represents both the shared nodes here in purple, if we combine these 2 sequences, and the unique nodes here in red and blue. And then doing analyses on this graph give you information on variation. A short while later, we were involved in a sort of brainstorming session of about a week at a sort of mini conference in the Netherlands on sequence-based pangenomics, and that resulted in a paper. This was a group of people with similar ideas. And so we wrote a sort of white paper where we said, hey, but there's a lot of opportunity here to do interesting things on large collections of genomes at the sequence level. So this could have different applications. For example, compression. If we store 100,000 E. coli genomes, maybe we don't need to store 100,000 individual genomes, but we can just store the backbone and then some variation on top of that. But we can also use it for visualization, for annotation, etc. And as I've said before, I'll stress it again, there's nothing new. It's basically still a road, but we basically want to scale up the type of analysis as is illustrated by this figure here. Top left, top right are still both means to get you from A to B, but the bottom right version likely does it a bit quicker than the old version on the top left. Now, while discussing this with this group in this workshop, we quickly figured out that different applications demand different data structures. So for some questions, maybe you can get away with just the unaligned sequences if you have different genomes. For other applications, you may want to do a multiple sequence alignment, where you still keep the original sequences, but you make clear where they align and where they have unique sets of sequences. You can also represent it in graphs like the brand graphs used in assembly, or sequence graphs, cyclic or acyclic. So here you would represent this repeat here. I think you would here have a repeat twice, in this acyclic sequence graph, but in the cyclic sequence graph you would represent it just once. So you would say here's this genome here takes a route back and then goes once more through this sequence region and then continues here. One maybe well-known example is the variant graph, and that's really tailored to storing and querying variation in human genomes. And specific, you can imagine that if you have a genome so two genomes, or a genome and resequencing data, which are 99.9% similar, then it makes sense to store just a sort of backbone with just the variations on top of that. So the variant graph format is really based on storing a reference and then some variations on top. In many of the problems we work with, we encounter slightly more complex data sets. So plant genomes are very complex. They're very often very highly repetitive. Some plant genomes consist of over 70% of repeats. They can be highly heterozygous, the chromosomes. There's polyploidy in many interesting crops. A potato has 4 copies of each chromosome. Strawberry, I think, has 8 to 12 copies of a chromosome. Wheat has 6 copies. So that makes life more difficult. And there's lots of varieties that people want to work with that actually differ quite a lot in their genome. Often also based on ancient genome duplications. So even in species with just a single genome or a diploid genome, we still find remnants of ancient duplications, which make life a bit harder. So we set out to create a pan-genome representation for plants, and I won't take you all the details here, but basically what we set out to do was that it would allow us to compress multiple full genomes without loss, which would sort of scale to the data sets that we want to work with, so hundreds of plants or animals, and that allow us to integrate all kinds of data that is useful for analysis. And this is sort of unique, I think, compared to other approaches. There's many approaches now to pangenomics, and some are very efficient and scalable. But often these use highly specialized data structures to which is very hard to connect other types of information. Yeah, so I think I'll skip the next slide. So what we came up with is that we represent the sequence at the basis of the pangenome as a colored compressed de Bruijn graph. I will show in a minute what that is. And then we have a K-mer index next to that to access the pangenome, and all kinds of annotation on top. And we store this in a graph database like used for social networks, for example. We chose for Neo4j. And so most of our algorithms are disk-based, which makes it very slow, but we use sort of standard techniques, and we hope we're now actually looking into also standard techniques of speeding this up by working in memory and also having distributed databases and parallel processing. So this is a small illustration of such a pangenome. It's actually 2 viral genomes that are stored here. You can see the sequences here in green. So every time you see one node, that means that a piece of sequence is shared. And such a bubble is where the 2 genomes have different sequences. So maybe a SNP or a small indel. And then you have here a blue node and a pink node, which indicate an organism, so basically a genome and a chromosome. Well, that's just one chromosome, so the start of the genome is also the start of the chromosome. And then there's genes and homology groups indicated. So you see here at each point 2 genes are found for the 2 different viruses, but they belong to the same homology group. So this is sort of like the core representation. Since then, we've been adding a lot of different annotation on top. So we, and I don't think this is even the full current picture, but we can go down to the sequence level, but we can also easily incorporate all kinds of annotations like phenotypes, like RNA expression, like gene structural annotation, and so on. So I would like to show you a couple of examples of what we do with that, about how to construct them, how we can then retrieve sequences, do structure queries, and then a couple of things we've been developing since on detecting orthology by including the proteome layer, doing read mapping, and recently also doing machine learning based prediction on these graphs. But we're still working on a number of different applications that allow us really to replace sort of standard analyses by operations on this pan-genome. And for this, we also collaborate with a couple of plant breeding companies in the Netherlands that are fairly interested in having such solutions in their day-to-day operations. All right, so the construction is fairly straightforward. It's an order n algorithm that basically walks over the individual genomes and constructs the database on the fly. So it maintains and builds up a k-mer index. The k-mer index points into the pan-genome graph. So, for example, if we have a certain k-mer here, ARA, then it knows that it's stored here in node 2, starting at position 1. If it's 0 based, so this would be position 0, this would be position 1. So the index immediately takes you there. So if you walk along the sequence, you can see whether a node is already there. If not, you add it, and then you add it also to the index, and you add a node. And sometimes that means if you find a certain k-mer here, ARA, for example, and you find that the next one is RAA, then you realize that you will have to split this node into 2 and create 2 new paths. So then you do a local operation in the graph to expand the graph. So that's not rocket science, but well, still tricky to get working correctly. And then, 2 sequences like this would in the end be represented in this compacted color-to-brine graph like this. So the colors indicate the sequences. We have 2 sequences, 1 and 2. The red nodes are the nodes, the sequences unique to sequence 1. The blue nodes unique to sequence 2, and the purple nodes are shared between sequence 1 and 2. And you see that also the reverse complement is taken into account. For example, here this last stretch of sequence 2 here goes into the k-mer TTT, which is a reverse complement of AAA. So actually loops back here to this node with a different kind of edge. So these are bidirected edges. So far nothing majorly complicated. Yeah, so you can already see that the graph represents variation. So a bubble like that corresponds to a SNP or an indel. And this is the story I told about the reverse complement. So this was sort of the basic approach that we set up to be able to store and query genomes. And we were able to, using some tricks, to be able to get it working on reasonable sized data sets in reasonable amounts of time. This is still nothing great. So I think there's now tools around that do 7 human genomes in a matter of half an hour or so. But again, these are highly specialized and would not be easy to get to work on a graph database like this. So we're looking more into what's technical solutions to speed this up than really dramatic new algorithms. So we're able to put together a pan-genome for, say, 20 plant genomes in about an hour. And this is then the maximum memory use. 90 yeast genomes in half an hour, and 60 E. coli in 20 minutes. Well, then we can do standard stuff like extract gene annotations or gene sequences or even full genomes. And we can do that reasonably quickly. Maybe not as quickly as just reading it out of a text file, a well-indexed text file, but it's fast enough to be workable, I think. One interesting thing that we get from working with this graph database is that we also have structure queries. So Neo4j comes with a language called Cypher. This is a bit like SQL, but it allows you to query graph structures. And so where, as I said, SNPs and small indels correspond to bubbles in the graph, you can easily do queries for such bubbles. For example, here, if you use this query, we say, well, match a node, which is connected through another node to a final node, so node N and M, and then connected back through another path, through another node, again back to N. So this basically says, look for a path of length 2 from N to M, and then a similar length path back. And then you return how often you find those, and you do this on the 3 yeast genomes. There's a small pan genome of 3 yeast genomes. Then we find that in 6 seconds, we find all 121,000 occurrences where there's a bubble of this size. Well, these are not all SNPs, because we didn't specify here that the length has to be exactly, that they need to differ by just 1. So if we add that, then it takes a bit longer and we find fewer SNPs, but still this is, I think, pretty impressive to see that you can get all the variation with just a single query. And so there's more of these types of queries that you can easily do, like finding sequences that occur in all genomes or sequences that are unique to one genome, etc. All right, given time, I think I'll skip one discussion that's on Arthology Detection. We have a paper about that in BMC Bioinformatics. We basically added a pan-proteome layer there and used the same tricks, a K-mer index, to basically circumvent having to do an all versus all comparison. And we could then detect orthology in a fraction of the time that you would need using standard tools at reasonable accuracy. So that really works. A similar approach we took in read mapping. So here, a problem is that if you align reads to a reference genome and the reads from which you sample a genome, the sample genome has a unique insertion or maybe some unique variants, then it's always a question of whether you can find this back. So maybe you're able to map some of the reads, but you're left with a bunch of unmapped reads that either are in regions that do not occur in your reference genome or that show too much variation. And again, in humans, this is often not so much of a problem. Humans do not often contain a lot of unique sequence, but in plants and fungi, this happens all over the place. A second motivation is to avoid redundant mapping, and this is more, again, from a computational point of view, that if you have a genome, a pan-genome of a couple of hundred species that are pretty similar, you would want to avoid mapping your reads against every genome all in the same vein. So a relatively straightforward read mapping approach that we came up with is to use the K-mer index. So we sample K-mers from sequencing reads, we look them up in the index. We determine candidate hits by clustering start positions. So as soon as we find that a number of start positions for the read sort of agree with each other, then we perform a full Smith-Waterman alignment. But if we've already seen it, so we sort of keep track of what has already been mapped, then we just copy the mapping. And so that saves a lot of time. So if we do this for genome 1, then and we encounter the same node where we find that in the same node also genome 2 goes through that node and has the same coordinates, then we basically just copy the mapping. And as an example here, we mapped here, I think this was, yeast genomes and Arabidopsis genomes. So these are 90 genomes of yeast and 19 Arabidopsis, 19 genomes of Arabidopsis, our model plant. And we're able to, so this is the time it takes per genome to map a 10x Illumina read data set on 8 cores. So you see that in the beginning, the actual algorithm is not as good as optimized, highly optimized algorithms like Bowtie, BWA, etc. But once the number of genomes increase, the time per genomes actually bottoms out to be much less. So this is, we win time here by doing this for all genomes at once. And also it allows us to find interesting new variation. And again, the paper, which is on bioRxiv at the moment, goes into more detail about this. A thing we've recently been working on is to do machine learning based on these pan-genome graphs, where the idea is that, again, this is nothing new. The machine learning itself is nothing new, but this graph gives us sort of easy access to lots of relevant information in one go. So we can do one of these cipher queries to get out a data set and transform that data set into something that can easily be used, for example, in scikit-learn. Do predictions, and then actually in the end we would also like to augment the graph again, ideally in an automatic way, with predicted edges and predicted nodes. So you can imagine that if you run this once, then you can, next to existing gene ontology annotation, you can also add predicted gene ontology annotation. So here's an example. We tried this on a pan-genome of over 200 Pectobacterian genomes. These are plant pathogens, so they're quite of interest. We know for 13 of them that they're virulent, and 24 that they're avirulent, and the 67 unknown. And we were actually able to, based on PFEM domains and sequence variation, I think, extracted from this graph, to predict this phenotype almost perfectly. But to show you, to sort of demonstrate how this could work, here's an example of where we try to predict the gene ontology term associated with a certain transcript by using PFEM domains associated with a transcript of a protein that interacts with, oh, sorry, I should point here, then maybe clear. Yeah, so the PFEM domain associated with a transcript of a protein that interacts with the transcript that has our go term. And so this is complex, but so imagine we think that related, so interacting proteins might tell us something about the function of our gene. So we can actually come up with a query that finds all interacting proteins and then find their PFEM domains. And that this is actually a query that does that. It's fairly straightforward. And then we're able to predict, so in the order of, this is an AUC of 0.9 or so for predicting gene ontology terms. So this is, I think, again, this is nothing that couldn't be done before, but then at least you would have to combine a lot of different databases and do a lot of groundwork before you could get here. And now it's fairly straightforward to develop a query like this and just add predicted edges. All right, so we're currently working on extending this, so also mapping RNA-seq data, calling variants, doing structural variation annotation. Maybe more interesting, also look further back in evolution, for which we would need notions of biological similarity or homology over longer evolutionary periods. So this is really interesting work and challenging work still going on. Given the time, I think maybe it's good to answer any questions that may be there now on pan-genomes. If not, then I'll continue with the second story. I don't see anything in the chat yet, but maybe if you have a question, now's the time to raise it.
Yeah, I think there are no questions in the chat, but if there are some, please raise your hand or let us know. In the meanwhile, maybe you can, because while you were talking, I was thinking about, you were describing the Neo4j graph with the sequences, and I was thinking about phenotypes, but then in these last examples, you seem to have phenotypes already in the graph. So maybe if you can explain a bit better how, so the Neo4j graph has all these kinds of nodes that allow you to do these things.
Yeah, so the graph database itself is fairly straightforward. It just contains nodes and edges, and you can assign identifiers to nodes and edges. And so we just add, like here, we have this genome node, which then points, which has one or more sequences, which has nucleotides and so on. And then we can just add a node phenotype and have create an edge between genome and phenotype. And that then we can say, give us all genomes that have this phenotype, or give us all the genes in genomes that have this phenotype, and so on.
So the connection between the phenotype is to the genome, or you can put the connection from different variants to the phenotypes?
So that's a good question. At the moment, we just assign phenotypes to genomes because we still sort of mainly work from the assumption that we have full genomes. And so one of the challenges in working with variants is that we don't want to store these all as individual genomes. We don't want to create, if we have 3,000 resequenced genomes, we don't want to create 3,000 full genomes and then store them, but to still annotate these types of things. Yeah, that's a good question. So then we would need something like a different node, maybe resequenced genome that could still be connected to phenotype and then point to individual variants in the graph.
Yeah, because what I was thinking is if you have a variation that changes the phenotype, if you have a way to put that exception without creating redundancy, of course, but if you have a variant where there is a protein that doesn't have the same function because it's a different protein, something like that. I don't know if you have a scheme to try to.
No, I don't think we've tried that yet. So we've been mainly been looking at genes and proteins in terms of homology groups and similar function, but we have not yet gone as far to, and we've coupled variants directly to phenotypes, but not through predictions of what effect it would have on the protein function. That's an interesting challenge tool. Yeah.
Okay, and I think we have a question here. So Ruben was asking do you have those k-mers, the k-mers associated for each genome only, or do you have more relations for each k-mer? I don't know if you can understand.
I'm not sure I fully get it. Maybe Ruben, you can ask your question, or
yeah, maybe Ruben. Yeah, you can ask it.
Can you hear me?
Yes.
Yeah, go ahead. Yeah.
Okay, so do you have the chimer that is a node that you represent from genome, correct?
Mm-hmm.
So, for instance, let's see your gene is a set of chimers, correct?
Yes.
More or less. So you have it directly represented in the graph, or you only have like the genome in the middle, in the node middle between the genes and the chimers?
No, so all k-mers are represented in the graph, but not, so it's a compacted Brown graph. So if these genomes are very similar, you would get one node with a long sequence, but all the k-mers are in there and indexed.
Okay, but I'm asking because, for instance, you can have several sets of chimers that are associated for one or more genes, and those genes are from different genomes, for instance. And that's why I'm asking if you have relationships directly associated with the chimer that are connected with one or more genomes, for instance.
Yeah. So here, so this representation does not do full justice. So here, when it says genome, there's multiple of these genome nodes.
Yeah.
And each genome can point to multiple genes, and these genes point to start and end k-mers.
Okay.
So it's really sort of a hierarchical system, and you can trace each genome as a path through the sequence graph.
Okay.
Does that help?
Yes.
Okay.
Yeah, so maybe you can go to the second part, so we don't delay this too much.
I'll do that, and I'll try to keep it short because I've been too optimistic again, I think. This part is about it's a completely different topic, but it's quite recent, and I'm fairly excited by it. This is about the idea of using protein structure for predicting certain characteristics of proteins, like protein function or interaction or enzymatic activity, things that are traditionally sequence-based. I think we're now getting to the point that we do have a lot of structures, maybe not all fully independent ones, but this graph here shows the growth of the protein database over the last years. We now have over 160,000 structures. I think something like over 50,000 unique ones. As you may have followed recently, people now consider protein structure prediction almost a solved problem, which doesn't mean that it's easily solved. So AlphaFold 2 has really broken all records and is getting close to well, near optimal performance. You still need quite a lot of heavy computer hardware and a couple of weeks of waiting time to solve a structure, but this is starting to be solved. So I think we can, in bioinformatics, we can start to think about how we can use structure more to predict things. And a challenge here is, again, this is also from a collaboration, is that we work with people who are interested in Sesqui terpene synthesis. I see I misspelled it there at the top. So these are small compounds that are produced by plants, and there's many different varieties of them. And so the Sesqui terpene means that it's C15H24. There's also monoterpenes and diterpenes, etc., that have different combinations of these carbons and hydrogens. But these Sesqui terpenes are interesting because they end up a lot in food as flavor, fragrance, etc. So limonene, the citrus, the stuff that you find in citrus peel, that is a Sesqui terpene. And that is used quite heavily in soft drinks. So maybe one drop of this stuff goes into a bottle of 7-Up. Pinene, that's the smell of certain trees that we use in toilet air fresheners. But also peppercorns, lavender, lemongrass, orange peel, etc. They're all different varieties of these Sesqui terpenes. And so we work with companies that actually want to understand how these compounds are created in plants, and then maybe transform this into a biotechnological process. So be able to produce citrus flavor or other types of flavors using bacteria. Now, a problem here is that these there's many different reaction paths. There's many different products. There's only little data available for each product, and we have even these enzymes, these Sesqui terpene synthases that can produce multiple different compounds at once. So that is really a challenge. And so we worked with the first thing we did was collect a database of all the known Sesqui terpene syntheses. This was around, I think we now have around 300 curated Sesqui terpene syntheses and another 6,000 terpene synthase-like sequences that we can place somewhere based on prediction, but that have not been curated, and that show really a large variety of sequence, but mostly also with a strong evolutionary signal. So we cannot easily compare sequences because they look more like the plants they came from than that they show similarity between enzymes that do the same thing. So as a first step, we wanted to see whether we can use structure to predict whether such a synthase makes products based on the farnesyl cation or the neurolyl cation. These are two different starting points, here shown on the red side and the blue side, that really that lead to a whole different set of possible compounds. And it's maybe not, it doesn't solve the entire problem, but it's already really worthwhile to know the cation that is produced, because it really narrows down later prediction problems. So how did we go about this? Well, we took structures. If we didn't have them, we used homology modeling. Then we extracted structures locally about secondary structure, about angles, about certain properties, surface properties, etc. And then try to use that in the machine learning approach to predict enzymatic activity of these Sesqui terpene syntheses. Now, problems that we ran into was that it's not easy to align structures. So if you want to do machine learning, you have to make sure that you use the same feature columns represent the same thing. So here you would need to do multiple structure alignment. That's costly, and it's often not very, these are not designed for doing machine learning, but rather for learning about the structure. And there's sort of lack of an invariant structure representation. So rotation and translation invariant. So we try to solve both problems at once by coming up with a representation called geometricus, which describes a structure instead of by k-mers, which is inspired by the sequence world, by shape-mers, which describes substructures. And we describe these local structures using 4 moment invariants, which are inspired by computer vision. I don't know whether it rings a bell with you, but in computer vision, for object recognition, people come up with these moment invariants, so numbers basically that describe an object like this one, no matter how you turn it or how you translate it, that the numbers stay the same. So they will be invariant to rotation, translation, and sometimes even scale. So we calculate these moment invariants for short substructures. We log transform them, we multiply them by a resolution m, and we discretize them, and that's sort of illustrated here. You see that these three structures, substructures, all end up with the same moment invariance, and then after taking the logarithm and discretizing, we end up with this shape mirror. So this shape mirror represents all these three structures. Similarly, this shape mirror represents these three structures. So I think you sort of get the point. The resolution is there to gives us sort of a way to tune the level of detail. You can imagine that if we multiply this here, so now there's a multiplication of 1. If we multiply by 10, then we get here 50, 0, 0, 0, but we could keep it apart from 51, 0, 0, 0. So the resolution sort of decides how extensive our feature set will be, but also the level of detail which we can keep things apart. Well, if we apply that to the entire protein database, I should mention we have two ways of representing substructures, either by following the sequence through the structure, or by just looking at a sort of ball around a certain residue, a radius-based approach. Then we actually find that a distribution like this, which you could sort of expect, there's a number of shape mirrors which occur in many proteins, and then there's a lot of these shape mirrors that occur in just a few proteins. Here's an example of one that occurs a lot, and not surprisingly, a substructure describing a helix, you find that 1 million times across many proteins. And we found that substructures chosen based on sequence actually generally work better than those chosen on radius. Where we had the idea that this radius would allow you to combine structures that are far apart in sequence, that maybe had folded back on each other. But in most applications, it turned out it doesn't help so much. Well, then a very simple representation of structure is then to basically just fragment two proteins or many proteins, calculate these shape mirrors, and then count how often we find certain shape mirrors. So this gives us a sort of shape mirror fingerprint, and we can then compare these fingerprints or calculate the distance measure between these fingerprints to get a distance measure in terms of protein structure. And we can do this fairly quickly, about 50 milliseconds per protein. So we have sort of a very cheap alignment-free structural similarity method. Well, we showed that this worked, for example, by doing a t-SNE embedding of a large number of kinases, and these nicely cluster into the families that you would expect. And they do this better than based on sequence, so this helps. We also showed that you can do this to sort of interpret machine learning applications. This was a very simple application in which we try to predict subtypes of a certain family of kinases. Based on structure, we could do that pretty accurately, and then you can go back and look which structure features, which shape mirrors are now actually important for this prediction, and then you find there's short, small subshapes here that either make a large difference between the classes. So whether they're present or not, this sort of short helix here is really predictive of having this P38 subfamily, and this short helix over here is really predictive of having an ERK and ERK type protein. Okay, now I think I should skip some steps because this is too much detail for now and time is running out. But we also, let me just mention, we also use this as the basis for a multiple structure alignment algorithm called Caretta, which is very much faster than existing algorithms and about as accurate. But then back to our prediction, we recently had a paper accepted in PLOS Computational Biology in which we showed that we could actually now predict reasonably well this main distinction between these two cations. And now it really depends how you split the data. If you, because the data is so biased, if you split the data randomly then you can do pretty well, but then you will have a lot of very similar sequences from similar species. So in the most extreme case, where you split the data according to clades, so you make sure that there's no evolutionary signal left between the train and the test set, then you do much worse, but still you get to an accuracy of 0.64, which is not ideal, but it's a starting point for further analysis. And it also helps people in practice if they can limit the number of proteins they have to test in the lab. We're currently expanding this, taking the next step, so moving to actual product prediction. And here we take a step, take the approach of not predicting just for the enzyme what compound it predicts, but predicting for enzyme compound pairs whether they react or not. So the problem has now become, given a protein and a compound, can we predict whether they react yes or no? And the first signs are that this works to some extent. So we get a higher accuracy than if we just use sequence. And we do find some structure fragments in the proteins and in the compounds that indicate what types of cations they work on and where the active sites are. And we're currently, with our experimental colleagues, testing this to see whether we can characterize new sequences. So we've identified a number of sequences that we want to have tested. And we're also creating a couple of mutants based on what we learned here to see whether we can then change the performance of an enzyme, so change what it makes or how much it makes of a certain compound. Okay, that brings me to my last slide. The acknowledgements, the work on the pan-genomes was mostly done by Siavas Sherkizadeh, who's now left the university and works at a plant breeding company. And is currently being continued by Eve Jonk here, and they were both co-supervised by Sandra Smit. And Sandra is really taking care of is supervising Eve, and many other collaborators, PhD students, master students, and other PIs involved in this process. Too much to list all of them. And the work on the protein structure, Coretta and Geometricus, was mostly done by Janani and Mehmet here, and co-supervised by Altjan van Dijk. So with that, I'm happy to take any questions, either on the protein structure prediction, protein structure features, or still on the pangenomics if they're there. Thanks.
Okay. Thank you, Dick. I think it's some impressive work. This last part of the paper on the machine learning prediction, you have that available, because we have also some packages with protein classification with different features, so it could be interesting to maybe compare those two approaches.
Sure. Yeah. So the plus computational biology paper has just been accepted, but that is mostly about the application. The Geometricus and Coretta method papers have been published in bioinformatics and what was it, computational structural biotechnology last year both. So if you look for this name, Dugai Raj, then you'll find the papers, I think.
Okay. And do you have this available as a software that people can use to different problems?
Yes, the software should be available on GitHub. And for Coretta, so there's multiple sequence alignment that I didn't discuss much. We also have a server application that you can use. So you can upload some sequences and then you can get a multiple structure alignment.
Okay.
But for both, the code should be available. Yeah.
I think we still had one question from João Sequeira on the first part, I think. Maybe
how is a pangenome converted into a GSMM?
Yeah, I think this relates to genome scale models. I think you have also worked on that in the past. Maybe João can explain a bit what he meant.
Yeah, so I wanted to understand how a pangenome that has all the variant callings that you resolve on the sequence alignment, how that is converted into a metabolic model. If that can be converted to a metabolic model that considers those variants in the proteins.
Yeah, I think that's a very good question, and we haven't reached that point. I think it's similar to what Miguel was just asking, right? How can we link variants in specific proteins to specific phenotypes? And I don't think we're there yet. So we certainly have no functionality yet to convert things into a genome scale metabolic model. And we can identify SNPs that could be used to that could be potential leads for phenotypes, but we're not at the point yet that we can easily use the pangenome to then explain how it leads to variation in proteins and how that would change functionality like in metabolic pathways, which could result in this phenotype. I do think the information it should not be hard to put the information in there or to query it out of there, but we just haven't worked on it a lot yet. Another reason is, and that's maybe not very nice for you to hear, but I think you guys working on these metabolic models, that you're really used to thinking about the whole cascade between a SNP and a phenotype. But especially people working in plants and animals, they have not considered this for very long. So they're very used to just finding a SNP and relating it immediately to a phenotype without caring what happens in between.
Because the alteration in the protein could change the activity of other proteins, right? Different metabolic.
Right, so your question is very relevant, but yeah, we haven't gotten around to that yet. No.
Thank you.
So I think you had another one on the second part, right?
Yeah.
Yes, about the specific identification of a protein, because I had a work where I was trying to identify isoprene synthases, and I could not understand how I could distinguish between the isoprene synthase and other types of terpene synthase, because in the identifications, for example, with the BLAST and other sequence alignments, they are always identified as such, because, as you said, there's so much identity in the structure. It's more about organism than about function.
Yeah. Yeah. So your question, when I read your question specifically, when is it enough? That I really don't know. So we try to come up. So we've now shown that structure gives us a bit more confidence in our predictions than just sequence. We're now working on this structure compound prediction. We're really trying to phrase that in terms of this approach, in terms of a ranked list of predictions with some confidence value attached. And now, yeah, I think the proof of the pudding will be that we send our collaborators into the lab to measure a number of sequences, and if they all fail, then we will be in trouble. But we don't at the moment have a good idea of where the cutoff is, or where sort of an absolute value is, where you could say, well, this we trust, this we don't.
You always have to go to the lab, right? Experiment.
Yeah. I always tell my students that bioinformatics is, in this case, can never predict the truth. It can only give you leads, and I think your systems biologist, so you realize this, that in the end, we always need to verify things in the lab.
Thank you.
I think that's a good. I think that's a good message, since we have lots of master in bioinformatics students here today seeing this presentation. I don't know if there are more questions. I think the first one for the genome scale models, maybe it's a good way for us to start collaborating, because I think it could be really interesting to go from a pan genome to a pan model of a set of sequences and of strains or species.
It would be interesting. Maybe at some point we can develop a master student project together. We have a number of master students here. I guess you have them as well. So we're happy to collaborate on supervision there.
That's a good thing to discuss in the next weeks. I don't know if there are more questions. If someone wants to ask anything, just put in the chat or raise a hand.
I think we're already over time, so maybe people want to go for lunch.
Yeah, I think since we are all at home, this is more flexible than usual. If we were at the lab at 12, everyone was rushing to the canteen. Okay, I think there's one question from Lucas. So Lucas, you want to ask the question yourself?
Hello, everyone. Can you hear me?
Yes.
Hello, Professor Ryder. My idea is about What do you think about the RNA sequence data? How does the construction of epidemiologic graphs, because the sequence, the RNA-seq sequence can contribute with great bubbles and alternative splicing, for example. Your idea is the use of RNA-seq data for feature or construct the genomic graph?
Yeah, a very good question. I wasn't clear about that. So what we have at the moment, when we say we store RNA-seq data or expression data, we are thinking about just storing expression values in some way, so that we can query, say, give me all the genes that have this SNP and that have a high expression in this genome, something like that. But we also want to develop methodology for aligning RNA-seq reads to the pan-genome, basically to do read mapping, not on one genome at once, but on many genomes at once. Then in that case, I don't think we'll add them to the pan-genome graph, because I think you're completely right, that will become very messy very quickly. And we would actually get a sort of very complex assembly graph in our pan-genome, which I don't think will tell us a lot.
Okay, thanks.
Thank you.
So I think we won't take more of your time. I think it was a very nice presentation and very interesting work. I have taken many notes to try to explore the papers in the next days, because I think there are many connections with things that we are doing, probably more at this second part, but also as we have seen also in the first. So I think we can, for sure, we will continue to collaborate. We thank you for your time, and let's hope that this collaboration can be more in a more presential way. We'll be glad to have you here when things allow in person.
I will be glad to come over. So thanks again for the invitation, and yeah, hope to speak to you soon again.
Okay.
So thanks to the audience for listening.
Okay, so thank you, Dick, and thank you everyone for participating. We have been above 50 participants the whole time, so it's a very good number. So we'll resume these bioinformatics seminars, I hope, in next month with another guest. So thank you, everyone. Okay. Thank you, Dick, again.
Thank you. Bye-bye.
Bye.